So these spiral-shaped bacteria that you see here under the microscope, they're not going to be very happy for long, because today I'm going to add a hand disinfectant to those bacteria, and I would like to see what happens to them. Well, I can already promise you right now, they're not going to be happy about that. They're going to die. We're going to watch how this actually happens, and then towards the end of this video, I'm also going to show you the effect of this hand sanitizer, this hand disinfectant on larger cells, like for example the ciliates, which were also present in the sample. But of course, I first have to say hi, hello, and welcome. Microbehunter here, Oliver here. Yes, I like to put things under the microscope, and today we're going to try something again a little bit more experimental. I would like to test the effect of this hand disinfectant, hand sanitizer on those spiral-shaped bacteria. But let's go back a little bit in time, and where did I even get those from? Well, I like to keep jars with pond water on my window so that I always have something to put under the microscope. And yeah, some time ago I actually did see that in one of those water samples there were many of these spiral-shaped bacteria. Evidently they reproduced quite well in one of those jars, feeding on decomposing plant and matter. Maybe also some tiny worms or snails that were present, provided sufficient food source for those bacteria, and they reproduced and multiplied, and basically here we are right now. So I decided, great, those spiral-shaped bacteria, because they move so quickly and rapidly, you would be able to see the effect of a disinfectant quite well. Do they stop moving? Do they run away from the disinfectant? How fast do they stop moving? How fast do they die? Well, a lot of questions that we can answer, and this is essentially what I've tried to do. Okay, so what I've done is I've taken a small sample, a small drop of this hand disinfectant, and I added it to the edge of the cover glass, and then capillary action has drawn in the disinfectant beneath the cover glass, and it got mixed in with the bacteria. And when we do that, we immediately see that many of the bacteria are swept away by the stream of the liquid, and this of course makes it quite difficult to observe. However, I could also see that some of the bacteria actually stayed connected to the glass surface. Evidently the distance between the cover glass and the slide was quite narrow, so many of them, or some of them at least, got sandwiched between those and could not move away. And when you looked at those, then you could immediately see, wow, they stopped moving, right? Essentially, there was an instantaneous death. The hand disinfectant immediately killed off those cells, and they stopped moving quite quickly. And also at the different places of the slide, when you checked those bacteria, then I could also see, all of them pretty much stopped moving. So essentially what I learned from that, this disinfectant works very quickly, right? It's an instantaneous removal of bacteria. Now, how do those disinfectants even work? Well, those disinfectants contain in many cases either alcohol and or hydrogen peroxide, and the activity of those two substances is a little bit different, because alcohol disrupts the cell membrane of the bacteria and of other cells, and this causes the cell membrane to burst open and the contents of the cell is spilled out. And hydrogen peroxide, on the other hand, is highly reactive. It produces highly reactive oxygen radicals, and they kind of react with pretty much anything that's organic. So essentially the hydrogen peroxide indiscriminately starts destroying molecules in the cell, and of course the cell is not able to survive that. So now what actually happens to the larger cells, like for example those ciliates over here? They are significantly larger, they're feeding on those tiny bacteria, but when they're exposed to the hand disinfectant, well, then they do not immediately recognize that the environment has changed. The reason I think is because it does take the disinfectant a little bit of time to actually diffuse into the cell. So first of all, this ciliate was happily moving around trying to feed off the bacteria. I think they're too large in any case, but I think that's a different topic. And when the disinfectant reaches those ciliates, then they start to slow down. So as the disinfectant starts to act, and then I could see that actually it stops moving, and then the cell burst open and the contents was spilled out. So you see that those cells too can be easily removed using hydrogen peroxide and or alcohol. Just wanted to share this with you. I hope it was interesting. I do invite you to subscribe to this channel if you like these type of videos. I wish you all the best. Happy microbe hunting as always, and see you in the next video. Bye bye.